Hey, this is Usman, and in this video, we'll try to explore one of the most essential and basic concepts in genetics. And hopefully this would also mark the beginning of a new Genetics 101 playlist focusing on all the relevant elements that revolve around the DNA, such as its structure, its discovery, and also its biochemical composition and the amazing biochemistry that is behind this molecule. And we'll also in further videos talk about how the DNA acts as the single unit of heredity that passes subsequently in every generation. So we'll cover all of that hopefully in the entire playlist. But in this video, I just want to talk about DNA, just introduce informally what DNA is, as well as what it actually is made up of. So just the introduction, basics, I would say. Intro and the basics, sort of the basic terminology that's related to DNA. And so that's what I'll hopefully cover in this video. Okay, so we know that DNA is a nucleic acid, and so DNA stands for Deoxyribonucleic Acid. And so the suffix nucleic acid would mean that it's actually one of the four essential biomolecules that help sustain life, because that's what a nucleic acid is. So I'm going to write that down. So DNA, or deoxyribonucleic acid, which is the full form of DNA. So it's a nucleic acid, and so nucleic acid. So if you're just, by the way, wondering what a nucleic acid is, they're actually one of the four essential biomolecules that help to sustain life, and they have many functions that help to do that. And nucleic acid, and there's actually four of them, and if I can just write that down quickly, that you have carbohydrates, proteins, you have lipids, lipids, and finally you have nucleic acids. So it's very essential, and it's very central to all forms of life, be it a virus or a complex organism like us, and nucleic acids. And so nucleic acid is actually where the genetic information is stored. And so that genetic information is used to actually perform all the cellular functions, to carry out all the metabolic reactions in the cell. And that genetic information, it's stored, it gets passed on subsequently from one generation to the next. And that's the nucleic acid, is where all the genetic code lies in. So nucleic acid, okay, so we know that it is a very essential molecule, and nucleic acid is a polymer of something we call a nucleotide. So nucleic acid, so we know this nucleic acid. So I'm just going to erase this. Nucleic acid is a polymer contains genetic info, and it's a polymer. It's a polymer of a nucleotide. Now I'll explain what a nucleotide is briefly. Nucleotide. Okay, and so because nucleic acid is just a polymer of nucleotide, it's also referred as a polynucleotide chain. A polynucleotide chain, because it's essentially just the repeated units, it's just composed of the repeated units of a nucleotide. And if you're wondering what a nucleotide is, Well, a nucleotide is the basic building block of a nucleic acid. And in this case, we would call it DNA. So since we know this is the structure of DNA, and this is DNA is a nucleic acid, so the repeating monomer, this basic unit that actually makes up all of the nucleic acid. So this one unit, so sort of like this, is a nucleotide. This is one nucleotide, because you can see in a DNA model or a picture of DNA that there's always one unit that's getting repeated, and that repeating unit is a nucleotide. So it's the basic building block of a nucleic acid and of DNA, because DNA is a nucleic acid. And I'll go more into what a nucleotide is made up of, because the interesting thing is, if we want to really understand the chemical structure of DNA, we actually have to understand the chemical structure of a nucleotide first. And that'll help us to then understand and imagine the molecular level, all of the nucleotides stranded together, how that would create a structure like this. So there are actually, so DNA is not alone. There's also another nucleic acid, and that's called RNA. So we have understood what a nucleic acid is. It's a polymer of nucleotides. Okay, so there's another nucleic, so we have nucleic acids, you have DNA, which is one nucleic acid, and there's another nucleic acid, and that's called RNA. And you have DNA here. And so DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. So ribonucleic acid. Ribonucleic acid. This stands for deoxyribonucleic acid. And so if you're wondering what the differences are between these two nucleic acids, they all actually differ both chemically as well as in their functions, I would say. So DNA is actually the ideal genetic material, or the ideal nucleic acid that we usually think of when you're referring to something being a nucleic acid. But there's also RNA, and that has different functions, and it also looks a bit different chemically. However, in this video, I just want to focus more on DNA. So we'll get back to RNA and its own entire story later in another video. But when we're talking about DNA especially, it's a polynucleotide chain. Both of them are polynucleotide chains because they're nucleic acids. And so it's a polynucleotide chain of what? DNA, it's a repeating chain of deoxyribonucleotides. To be more specific, because nucleotides are actually just, they can be, are you referring, when I say nucleotide, you might be confused, because am I referring to the nucleotides that make up this nucleic acid, or am I referring to the nucleotides that make up this nucleic acid? So to be more specific, we actually just prefix this deoxyribo in front of the nucleotide to say, because nucleotides actually differ in the chemical structure as well between the two different kinds of nucleic acids. So that's why we want to be more precise. And so ribonucleic acid, as you would guess, it's composed of ribonucleotides, or ribonucleotides. So two kinds of nucleic acids, two kinds of nucleotides. And so now we have understood sort of the basic understanding of what DNA is chemically, let's talk more about the structure of a nucleotide. So I'll erase this and even the model, but it's very useful to actually be able to visualize the DNA molecule as we are also trying to explore more and unravel more of its mysterious looking double helix structure. So now we're going to talk about structure of a nucleotide. So I'm going to write that down. Structure of a nucleotide. Okay, so a nucleotide is just essentially just made up of 3 subunits or 3 parts. So nucleotide is made up of 3 parts. First, you have a nitrogenous base. Nitrogenous base. Second, you have sugar, a pentose sugar. So a pentose always refers to 5 carbon sugar because it's a sugar it's ending with an O's. And you have a phosphate group. And first, let's talk more about the nitrogenous base, and that's what we'll be talking more in detail in this video. So we'll first talk about nitrogenous bases, this video, and nitrogenous bases. So there are actually 4 essential nitrogenous bases in DNA, and there's also 4 found in RNA. However, the 4 that are found in DNA are not the exact same as the 4 found in RNA. And that's when I was talking about the chemical structural difference between RNA and DNA, or sort of the biochemical composition in that aspect. That's where the difference lies between the 2 nucleic acids. Okay, so you have 4 essential nitrogenous bases. And these nitrogenous bases, and I'm going to talk about those 4 are, but those nitrogenous bases, they essentially compose, they are the basic unit or the basic building block of the entire genetic sequence. So when we say what's your genetic sequence of what's the genetic sequence of an organism, we're talking about the combination of these 4 nitrogenous bases, how they are randomly combining together to form a long chain of pattern, or not a pattern, a long sequence, and that is what we refer as a genetic code. So that's what we're trying to refer to when someone's asking us what a genetic sequence is. Okay, so let's see. Okay, so it's very important. A nitrogenous base, as I said, it is sort of the basic unit of the genetic information, because the genetic information is actually because the genetic information is actually, when we talk about the genetic information, we're actually referring to the genetic sequence. That's how the information lies in the cell or in the nucleus, in sort of a sequence. So genetic information. And so, okay, so we understand that, but why does it say nitrogenous and why does it say base? Because if you have a good understanding of chemistry, any word that has base in it, which would refer to that chemical having basic properties. So, and it's true. So firstly, let's talk about nitrogenous. So nitrogenous bases, well, they're actually, they're fundamental carbon compounds, organic compounds. However, there is an adequate presence of nitrogen in it, and so we call those compounds nitrogenous compounds. So they have and once we'll draw the structures of each of the compounds and understand more of the sort of the biochemistry in that, we'll actually be able to understand why it's called nitrogenous. So there's an adequate presence of nitrogen, so N of nitrogen, okay? And it's called a base, so the last word, the base, because it's actually basic in nature. So it has basic properties, so it's basic in nature. And now you might be wondering, because DNA ends with an acid, the A in the DNA refers to an acid. So why would a nitrogenous base be basic, and how can that mean or conclude DNA being fundamentally acidic? The thing is, DNA as a whole is acidic. However, it's just one component or one part of the DNA which is actually basic. And once we'll talk more about the phosphate group, that's acidic. It's actually highly acidic. And so that DNA overall, once we actually refer to DNA, or once we're visualizing DNA as a whole molecule, not just as a part, we would call it as a whole acidic, because it shows acidic properties as a whole, but not as a part. As a part, the first part would be basic, the third part would be acidic. So as a whole, it's acidic. However, these bases are basic. And short form for nitrogenous base, we just call them bases. And so like I said, there's 4 of them in DNA. So in DNA there's 4 of them. So we have in DNA 4 bases. In RNA you have 4 bases. Okay, so what's the difference? Well, let's actually start naming those bases. So in DNA, you have first adenine, one of the nitrogenous bases, one of the most essential organic compounds. And adenine is not only found in the nucleus in the DNA molecule, it's also found in mitochondria. And actually, adenine is a very important composition of ATP. That's why we call that adenosine triphosphate. And we'll talk more about adenosine, what that means, and the role of adenine in synthesizing ATP. However, let's just talk about in terms of DNA right now, adenine, and we actually refer adenine as A. And then you have thymine, and that can be denoted by T. Cytosine, C, and guanine. So it's just the first letters that we denote these bases by. So A, T, C, G, adenine, thymine, cytosine, and guanine. Okay, so in RNA, you also have 4. And the interesting thing is they're so similar. They're actually 3/4 the same. It's just that, so you have adenine again. However, it's just that the thymine is replaced by another compound, another biological compound, and that is uracil. URACIL, denoted by U. And so uracil is present in RNA but not in DNA, and thymine is present in DNA but not in RNA. So once we are actually converting one nucleic acid into another, so from DNA to RNA, we not only just like change the structure of the function and all that, we're actually just changing the fundamental composition. So we're changing the thymine and converting into uracil, because the others are just the same. So you have cytosine here, and you have guanine as well. So it's just this that makes the entire difference. So it's just this. Uracil, okay. Now, So as a whole, you can say that there are 5 bases. However, not in one nucleic acid in overall, because the other 3 are just the same. Okay, so now we're gonna actually try to understand the chemical structure of these bases. But in order to understand that, first let's try to understand another concept, and that is a very, so I'm gonna erase this. It's a very important concept to understand. So we actually just classify nitrogenous bases into 2 categories. So categorize them. And those 2 categories are, so I'm gonna just classify these into 2 on the right. Okay, and so you have first purines and second pyrimidines. So by the way, these are not bases. We actually just classify all the 5 all the 5 bases into 2 categories. Okay, so purines, and what's the difference between purines and pyrimidines? Well, for purines, and they both are heterocyclic. So first, okay, so purines are heterocyclic compounds, organic compounds, and so purines have 2 carbon rings. So carbon rings. And pyrimidines, which are also heterocyclic compounds. So that's not really a difference, but I'm just writing there. But they have only 1 carbon ring. And there are names given to those rings. And I'm going to explain to you what that ring is. But the first is there are 2 rings. And so there's a 6-membered carbon which is attached to a 5-membered carbon compound. So you have a 6-membered carbon, and you will now be thinking what it looks like. It's just a hexagonal structure. So that 6-membered carbon attached to a 5-membered carbon compound, a pentagonal structure. So you actually have, the first one is called a pyrimidine ring. So the 6-membered carbon ring is called a pyrimidine ring. Pyrimidine ring. And pyrimidine ring is a 6-membered carbon compound. Then you have the 5-membered carbon compound, and imidazole ring. Imidazole ring, and it's 5. Okay, and so over here is just 1 carbon ring, and as you would be guessing, the name of that is just a pyrimidine ring. So purines are actually composed of pyrimidine rings, and so are pyrimidines. So pyrimidines compose of pyrimidine rings, and purines compose of 1 pyrimidine ring and 1 imidazole ring. Now, if we're talking about the nitrogen, the level of nitrogen in it, so purines have, so this is 1 difference. I'm gonna write here 1 difference. Okay, and now the second difference is that purines have 4 nitrogen atoms, and pyrimidines have 2 nitrogen atoms. So pyrimidines have 2, and those 2 nitrogen atoms are present in the pyrimidine ring. Every pyrimidine ring has 2 nitrogen atoms. Now, yes, you might be thinking, okay, so the pyrimidine ring over here as well should have 2 nitrogen atoms, and that's correct. So this has 2 nitrogen atoms, so 2N, and because I told you there are 4 in total, the imidazole ring would also have 2 nitrogen atoms. So there are 4 N atoms in 1 purine compound, and so 2N, just 1 ring, 2N or 2 nitrogen atoms present, and that makes a big difference. So these two differences can easily help us to identify or differentiate between a purine and a pyrimidine. Well, the first difference is actually more easy, because you just have to note if there's a 1 carbon ring compound, it's a pyrimidine, and if it's 2, it's a purine. So that's a difference. And then there are many other differences, such as the boiling point, the melting point, how they bond, and all of that. And so, but we'll not go more into that because these are just the starting differences. Now, so we got that clear. So now you might be wondering which base is which type of compound. So I'll explain that now. You have 5 bases for DNA, for an RNA, and adenine, so A, and guanine, G, are both purines, okay? And cytosine, okay, uracil, okay, and thymine are pyrimidines. So AG, purines, CUT, pyrimidines, okay? So now, if it's if it's kind of difficult for you to understand or to remember which base is purine or which base is a pyrimidine, there's actually a mnemonic, and that mnemonic is, right, so for purines, the mnemonic is pure as gold. Okay, so pure, so I'm gonna write that in red. Here's the mnemonic, pure. Okay, actually it's better if I color code the essential letters and the non-essential ones. So pure as gold. And this is just a mnemonic that's widely used everywhere. And so pure as gold. And because purines start with P, so purines, it's very similar to pure and as gold. So A for adenine and G for guanine. And the mnemonic for pyrimidines, you have cut the pi. So cut, or actually, cut the pi. And this pi is actually spelled as PY. So the thing is, C stands for cytosine, U for uracil, and T for thymine. So cut, that's what the word is from. And pi refers to the initials of pyrimidine. So that's a very common and a very simple mnemonic to identify which base is a purine and which one is a pyrimidine. Okay, so now that we have understood that bit of information, and by the way, when I was talking about the genetic sequence, I was talking about the genetic sequence of these letters. So the genetic sequence from, let's say, from one point to another point in a DNA can have the sequence or the random combination of letters, like you will have adenine in one nucleotide, thymine in another, cytosine in another, guanine in another, guanine again, cytosine, thymine, adenine, thymine, cytosine. And so once we figure out the nitrogenous base in every nucleotide that's below and above it, so we'll actually be able to form this sequence, and this sequence is our genetic code, and our genetic information is represented in this genetic code. Okay, so this was just a bit about that, but we'll actually try to explore more about this and all the other properties and features that revolve around the genetic sequence. And hopefully we'll also be talking about how to sequence a genome. So we have now purines and pyrimidines, okay? And so now let's try to understand the chemical structure of purines and pyrimidines, okay? And so we'll be drawing those carbon ring compounds for both purines as well as pyrimidines. Now here's the thing. If we talk about purines first, Purines, there are two kinds of purines, adenine and guanine, but they both differ slightly in their chemical structure. Like you have a functional group here but not there, and the positions are varying. However, what happens is that there happens to be a general structure of purines and a general structure of pyrimidines, and once we study specifically for every nitrogenous base, there are some variations, but there's a general structure that we can use to represent for any nitrogenous base. And I'm going to draw that for both, just a general structure. And then we'll also be trying to understand the numbering, how do we number the positions. And so yeah, so let's just start with that. And so I'm going to have so we know that purines have two rings. There's first you have a 6-membered pyrimidine ring. Okay, it's a bit irregular, but it's fine for right now. So, and you also have attached to the 6-membered, you have a 5-membered imidazole ring. So it's something like this. Let me make it a bit more regular, a regular pentagon. Yeah, it's fine. So, yeah, that's better. Okay, that's a pyrimidine. Sorry, that's a purine. And so purine has the two different rings. And so if I were to just right now quickly denote the rings, the names of the rings, it's a pyrimidine ring. Pyrimidine ring, imidazole, imidazole ring, and over here, when we're gonna draw the ring for pyrimidines, it's gonna be a pyrimidine ring, a 6-membered pyrimidine ring. So let's draw a 6-membered heterocyclic pyrimidine ring. Okay. And you have 3 pyrimidines and 2 purines. So this is a pyrimidine ring. Okay, yeah, you got that. So now we have the 2 rings, we have the 2 general structures. But now let's try in detail to actually understand the placement and the positions of the various elements and also the bonds and the functional groups. So here's the thing. Generally, once we draw a polynucleotide chain, we generally just draw this as the component of a nucleotide. Okay, generally. And the same thing goes for a structure of a sugar as well as the phosphate group. We're just generally showing the structure in forms of rings. So what I mean by that, I mean that we're not trying to actually label the various atoms and the bonds and show the double bonds and the triple bonds and all the other kinds of bonds as well as the functional groups. We're not showing them because for some reasons. Firstly, it makes things look very complicated because it's all so congested if we're drawing a polynucleotide chain. And so it's just for simplicity. It's all simple. It's more convenient. So that's why we're doing that. However, now that we are actually trying to study the chemical structure of all purines and all pyrimidines, we will go into details of all the functional groups and all the bonds and all the other atoms and their placements and the numbering and the conventions, everything. Okay, so let's start off with purines. So the general overview, the general structure that we have is this, and with the atoms, I'm going to label them. So first, let's talk about the nitrogen atoms, okay? And I'm going to do that in green. So purines, like we know, have 4 nitrogen atoms, and pyrimidines have 2 nitrogen atoms. And the pyrimidine ring in the purines have 2 nitrogen atoms. And the imidazole ring has also 2 nitrogen atoms. So, where are those nitrogen atoms located? Well, it's quite simple, because for a purine, in the pyrimidine ring, you have a nitrogen atom in this position at the bottom and the position over here in the upper half. Okay? And so that's the position of ring, or the pyrimidine ring, the nitrogen atoms. And because this is a pyrimidine ring, because this is a pyrimidine ring, and since these are the two positions of the nitrogen atoms, the same positions of the nitrogen atoms in a pyrimidine would be here, because all the placements and everything applies for a pyrimidine ring in a purine would also apply for a pyrimidine ring in a pyrimidine. So, okay, and so we're done with that. So now the nitrogen atoms in this 5-member heterocyclic compound, they are located top and down. So it's very simple. It's just top and down. So remember, bottom, upper half. So you know it's like up, top right. So bottom, top right, and bottom and top. And it's actually very simple now to understand, because remember, you have 2 on the top, 2 in the bottom. You don't have any in the over here or any over here in the middle. Okay, and for pyrimidine, if you understand the purine, where the nitrogen atoms are, especially in the pyrimidine ring, you technically, by definition, understand where the nitrogen atoms are in a purine. Okay, so now we're done with the nitrogens, and now let's fill the carbons. And I'm going to use the carbons with red. Remember, these are all organic compounds. They have to have carbons. Carbon is essential to all life. Okay, so let me just remove the vertices. So those vertices are talking about there's some atom generally, and we do that in organic chemistry for all compounds. So if you've ever seen someone drawing a soap molecule, they usually just draw a zigzag, but every vertex on that zigzag line talks about a CH2 or a CH2 compound. Okay, and so now we have the carbons over here in both pyrimidines as well as purines. Carbon, 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 carbon. Okay? Well, you know, pyrimidines don't have a ring attached, so it looks like it's at the end, but it's in the center for both. Okay, so now we have carbon over here, and carbon over here. So wherever there's an empty space and there's no nitrogen, so that's where carbons are. So carbons fill the rest. Okay, so these are this is the general structure, and now let's try to fill the bonds and the remaining, and whatever we can fill with a bond, we'll usually do that with a hydrogen atom. Okay, so the CHs and all that. So let's see, you have a double bond here. Yeah, you have a double bond. You have a double bond here, double bond here. So if you exclude the nitrogens, this actually looks like a benzene ring because of the alternating double bond. And it's a heterocyclic compound. So if it was only a homocyclic compound, it would look like benzene. Okay, and you have a double bond here. Okay, now let's first finish purine. So we know that carbon can bond at most, at least 4 it can have 4 bonds. So the carbon atom over here, it has 4 bonds. 4 bonds. Let's see, 2 and 1, 3. That means there must be a hydrogen over here. So let me do hydrogens in blue. So there must be a hydrogen. Okay, so nitrogen can have 3 bonds. So 3 carbon. Okay, so there must be a hydrogen as well. Okay, let's see. So we're done with the pyrimidine ring here. Yes, and let's see. The nitrogen has 3 bonds. The carbon, okay, so the carbon needs a hydrogen over here, so it can have the 4th bond attached to a hydrogen. over here, over here, yeah. And let's see, nitrogen 2, so it's a bond of hydrogen. Okay, so I'm assuming that is it for the purines. Let me just confirm once again. I won't be wrong. Yeah, I think that's it, yes. So we're done with the purine. Now let's move on to pyrimidine. So pyrimidine will be almost the same thing, except for in pyrimidines. So yeah, so in pyrimidines we also have the alternating double bonds in the pyrimidine ring, sorry. But remember, every pyrimidine, like a cytosine, thymine, as well as uracil, they have very different, quite different chemical structures of their own, but they follow this general rule of a pyrimidine. So the general rule of a pyrimidine, so you might be wondering, okay, so this is the general overview structure of a purine and a pyrimidine, but you're telling me that adenine, thymine, cytosine, guanine, uracil as well, and uracil, they all have their own structures. So why are we actually having a general structure over here? It's because in the general structure, then we know where the nitrogen atoms are and where the carbons are and where these all, so we know where the nitrogens and carbons are. It's just that sometimes you have the hydrogens that are replaced by another functional group. Okay, so a hydrogen can be replaced by, let's say, a methyl group or an amino group or a carbonyl group. Well, if it's a carbonyl group, you'll have a double bond, but just remember that it'll be somewhere like over here. Okay, and if this was carbon or something like that. Okay, so that's where the fluctuations are, and the bonds. There's also difference in the bonds. Like adenine has the alternating double bond, but guanine doesn't, and both are purines, so they should follow. But it's just carbons and the nitrogens which are placed there that is being followed in their own independent structures as well, in their own independent chemical diagrams. So let's see. Okay, so there's a double bond generally, again, general structure. Let's see. So I'm going to use blue for hydrogens now. So now let's see. This nitrogen over here, I think it's done because it has 3 bonds. Carbon needs a hydrogen, just like that. So this is done. Okay, I think it's another hydrogen. So those are bonded there. Needs a hydrogen as well. Okay. Okay. So, well, that hydrogen over there is replaced by this bond between the nitrogen in the imidazole ring. That's why you don't see one there. So I think that's it, and we have color coded the atoms, and the bonds are in black. So yeah, so now let's talk about the numbering, the positions. How do we number the positions of the various positions or the vertices on the pyrimidines and the purines? Right? Okay, so a very key note, essential point to make here. Once we're numbering them, once we're numbering these compounds, we would just number them as the position of this element, the position of this atom. So if you didn't understand, I'll just give an example. So let's say if this over here was the first position, which it actually is, but if this was the first position in the pyrimidine, we'll start with 1. So position 1, position 2, position 3, and position 4, and position 5. So we're not, because in some other compounds that you will encounter and learn about, we actually number the positions, and we denote that position by a prime. Okay, so it's usually an apostrophe, and we call that 1 prime. So when I'm talking about 1 prime, I'm talking about the position 1, but we're not going to prime them. So these are unprimed positions. Why? Because in the nucleotide, you have the sugar, like I told you, the pentose sugar. And once we're going to talk about the pentose sugar, we number them as well. And we number them with primes. And so not to get confused, because the sugars and the nitrogenous bases are bonding together. So we don't want to confuse with the numbering system of this as well as the numbering system of that. So here where these are unprimed positions. So we're not priming them. If you would if you have prime as a word, we're not priming them, and those are unprimed. So these are unprimed, sorry. And the ones in the sugar, the positions in the sugar are primed. So those have the apostrophes and so on. That's a very essential note. So these are not primed. So let's start. So in purine, so there's basic convention we have to follow for purines as well as pyrimidines. So in the convention system in purines is that you would have, we first have to, in the purine, identify the pyrimidine ring. So this is the pyrimidine ring. So we've done that. So the first step is to identify the pyrimidine ring. And once we identify the pyrimidine ring, we have to number from the nitrogen atom that's in the upper half, or the nitrogen atom that's above. Now look, there's only two nitrogen atoms, one's below, one's above, right? In the pyrimidine ring, this would be the nitrogen atom where we'll start the numbering from. So this is 1, not prime, just position 1, okay? So from that position, from this nitrogen, we start numbering the entire pyrimidine ring clockwise, okay? So we'll number them clockwise, like this. That's clockwise direction, and so that's 1, this would become 2, this would become 3, this would become 4, 5, 6, so on, okay? And this is, so for this, not to get confused, there's no nitrogen over there. Anyways, so there's 6, the 6th position. Now, it's very simple, this numbering convention. So we've reached the 6th position. So we've numbered all the 6 atoms in the 6-member pyrimidine ring. So now what we're doing is we're now going to go and identify the imidazole ring, okay? So we're going to move on to the imidazole ring. So we've ended our numbering here at this carbon hydrogen, and now we're going to move to the imidazole ring and start from this nitrogen, okay? So start from here. Now we start from the upper half, the nitrogen in the upper half of the imidazole ring, excuse me. Now it's very easy to understand which way you have to number, because you see we have already numbered this 4 and 5 positions in the pyrimidine ring, and we can't number them twice, right? So we can't now go clockwise, we have to go anti-clockwise. So we're going to go 8 and 9, and that's the positioning and the convention system that has been set up for numbering these. So here we go counterclockwise or anti-clockwise. So that's how we do it, and that applies to all adenine and guanine as well. Okay, so just to recap, we start from the nitrogen atom in the pyrimidine ring. So first step is to identify the pyrimidine ring, and then go in the pyrimidine ring, identify the nitrogen atom in the upper half, so the top one over here, and start numbering them clockwise. Okay, you number them clockwise. So we're numbering them clockwise. You end up at the top. Okay, so the 6th one, you move, shift left to the imidazole ring, and at that atom itself, so you're just shifting, so we're not moving down, or I'm just shifting left, you end up with a nitrogen atom, go anti-clockwise, because clockwise, if you go, you'll number them twice, these double bonded carbons. Okay, so that's we're done with the purines, and let's move ahead with the pyrimidines, okay? So with the pyrimidines, we're not going to start, because you might now think, oh, I said initially that the placement and everything, the rules that goes for a pyrimidine ring in a purine would be same for a pyrimidine ring in a pyrimidine compound. However, not the numbering. The numbering is a bit different, okay? So we won't start from this nitrogen atom. We're actually going to start from the bottom one. So yes, it's actually now. So we understand if it's telling it's telling us to number and the nitrogen atom in the upper half of a pyrimidine ring, we're definitely talking about the numbering system in the purine. If it's telling us, if there's like a question, a question is telling us to number the or if someone is telling us to number the nitrogen atom in the bottom half of the pyrimidine ring, we're talking about pyrimidines solely. So we start from this. This is our initial point 1. And now we, instead of going clockwise like we went here, we actually move anti-clockwise. We're numbering them anti-clockwise. So like this. Okay? That's how we're numbering them. So we're gonna go 1, and this is 2, 3, 4, 5, 6. And since this is just basic conventional rules that we have to follow, there's really no, you just have to remember these things, and you know, there's no shortcut to it. So that's it. We understand it's anti-clockwise. So start from the nitrogen atom in the bottom half, move anti-clockwise. Here we start from the nitrogen atom in the upper half and move clockwise. So it's opposite. It's the nitrogen atoms are opposite. The numbering rotation is opposite. Okay, so now we've understood a great detail of the these pyrimidines and purine structures. So I would like to just, you know, end about this discussion with a side note that in some diagrams we would see the pyrimidine or the purine flipped. So you will see the imidazole ring on the other side. So if I would, for example, just draw it over here, instead of seeing an imidazole ring first like this, sometimes we can see in some diagrams, you know, we'll be able to look, we'll be seeing the imidazole ring on the right side. Okay, and if that happens, if that is the case, the numbering system would switch. It would switch how the compound has rotated. So then we're gonna number, because remember, nothing's changing. We're not changing the pattern. We're not moving the 6 in there and here and there. We're not changing the position of the atoms. So because we're not changing the position of the atoms, we're just rotating the compound as a whole. The numbering system would just shift as a whole. So if it happens to be laterally inverse or laterally shifted, like it does in most diagrams, and I'll explain in a future video why that actually occurs. And it has something to do with the double helix, where one strand is opposite to the other. I'll talk about that later, but in detail. But so if that happens, the 7, 8, and the 9 positions of the imidazole ring would just be over here. No big change. It's just like that. Okay, and so that's how it is, and we would just, you know, it'll just be fine. We just have changed and laterally shifted, you know, but you'll never find something that's rotated 90 degrees. You won't find a purine rotated 90 degrees, so you won't have any weird looking shapes like this. You won't have a purine looking like this, okay? No, that won't happen. It'll just be laterally shifted. Or it may be a bit upside down, but no, not like in this type of rotation. Same thing goes for pyrimidines. So if you let's say, look, so the thing is, some pyrimidines would have, you can easily identify how the shifting is occurring because you have nitrogen down here. Now if you end up with a nitrogen at the bottom, but a nitrogen over here in the pyrimidine structure, okay, we know that it has been shifted, because we have learned that the nitrogen is always in the upper half. It's always in the upper half. Okay, so why am I showing you this? Well, this is the ideal convention. This is the ideal way to represent a purine and a pyrimidine. Why? Because I'll explain to you once we finish drawing the entire nucleotide chemical structure at the end of explaining about all these subunits of nucleotide. But I'm just going to tell you something quickly. The N, you have, well, the NH over here, okay? And this is actually supposed to be hydrogen over here. However, because this is a general structure, there's a double bond, and you know that varies in the other pyrimidines. So, you know, you won't necessarily have a double bond. Okay, So the thing is, there's an H in the cytosine, guanine, and thymine. So this NH is the attachment site of the nitrogenous base with the sugar in the nucleotide, the pentose sugar. Okay, so the pyrimidine, the pyrimidine and the purines are attaching on this site, and the attachment is usually, so if this is a pyrimidine and the purine, the sugar, which is the pentose, is going to be attached from this vertex to this vertex. So it's down, okay? That's why if we would have flipped it, the sugar should have been on this side, but the sugar is on the left side. That's why I'm drawing it in this sort of configuration. Okay, I'm not gonna laterally inverse it and show you. But yes, if we're talking about another strand, which is the opposite, it's the other way around. Okay, so we have got that cleared. And let's see, so we're done talking about purines and pyrimidines. And now let's try to just quickly draw the structures of the nitrogenous bases. And now that'll be very easy, but I'll be explaining to you while drawing about the differences between the various groups and bonds and everything in between the purines and between the pyrimidines. So let's start. Okay, so let's see, purines on the top and pyrimidines here. So we have lots of space to draw all the bases, and this is A. Let's see, we'll draw guanine over here, we'll draw thymine here, cytosine here, and uracil here. Okay, so let me just draw the general structure. The imidazole ring. Okay, bonded to a pyrimidine ring. Same thing goes here. Okay, for pyrimidines, so for pyrimidines you just have a pyrimidine ring. And so same thing for uracil. So thymine and uracil are both pyrimidine. And so I think I should draw the purines a bit bigger, but so it's fine actually. Okay, so like we said, nitrogens over here and here. Oh, and here and here. So there's nitrogens here. Nitrogen, and there's nitrogen. Just remove the spaces now for all, because anyways we have to. Okay, nitrogen over here, and nitrogen over here. Okay, and over here you have the same thing. Like this. The nitrogens are at position, in pyrimidines they're at position 1 and 3, remember. So now that we have learned the numbering system, we can also remember the placement of the various atoms, especially nitrogen, based upon the positions where they are. So nitrogens are usually placed at positions 1 and 3 in the, or sorry, not 1 and 3, actually it's right, yeah, they're placed 1 and 3, because I was thinking they go clockwise, but yeah, so 1 and 3 in pyrimidines, okay? And so 1, 2, 3, okay? In purines, they are placed nitrogens are placed in the pyrimidine ring at 1 and 3 again, and they're also placed in the imidazole ring at position 7 and 9. So 7, and that's 8 and 9. Okay, whoops. So yeah, so we're done with the nitrogens. Let's start with the carbons in red. So we know there's carbon, carbon, carbon here, carbon here, carbon, 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 carbon. Okay, so now when I was telling you initially about where the hydrogen gets substituted with another functional group, so now it's very important, and that's where the differences lie. Up until here, it's the same. The bonding and the positions of the functional groups, and the substitution of hydrogen with other functional groups is where the differences lie. So up until here, it's all the same. Okay, the general structure. Now let's talk about the specifics. So let's start with adenine, and so like I said, adenine has the double bond, the alternating benzene-like shape. But guanine, it actually just has 2 bonds. Oops. Okay, so 2 bonds, and they're, oh sorry, not beside, they're alternating. So yeah, okay, so they're alternating, but it doesn't have a third alternating bond here like adenine. That's one difference. Okay, so let's see what's another difference. Okay, so you have hydrogens over here, both, okay, and because we need another valency, I mean we have to fulfill the entire 4th valence electron that gets shared with the nitrogen on both sides. So now that's the same, there's no difference there. Let's see, yeah, you have the nitrogen and the hydrogen. The sugar nitrogenous base attachment site, that's the same. The NH is same. NH, and I'm not even gonna do here, but I'll first finish purines, then move on to pyrimidines. So NH, it's the same, that's for the sugar attachment site. So let's see, I think we are done with the bonding. So yeah, I think that's it for the hydrogens. Oops, there's a hydrogen over here actually. And yeah, so now let's talk about the differences here. Instead of the hydrogen, this is the second difference now. The hydrogen, instead of the hydrogen at position, what position is this? Position 2. Instead of the hydrogen at position 2 in adenine, well, in position 2 in guanine, you have not hydrogen, but you have an amino group. An amino group is NH2, so you have bonded to an NH2. And actually, let me just do the same thing, bonded with hydrogen. Okay, that's a big difference. That's the second difference. And there is an amino group here, but not at position 2 like it's in guanine. It's in position 6. NH2 amino group. Okay, and at position 6 in guanine, what's there? Well, you have a carbonyl group. And every time you see, and if we don't know there's a carbonyl group, but if you understand that there is only a single bond, so carbon has still 2 valence electrons left to share them with some other compound, and you know that NH2, the amino group, the methyl group, they all can just share 1 electron, they all can have 1 bond, but a carbonyl group can have 2 bonds. And so we would ultimately just by logic know that there's an oxygen there that has to share its 2 electrons with the remaining 2 valence electrons of carbon. Okay, so there's 3 differences now. The first is that we have the bondings. There's an alternating bonding like benzene-like bonding. Over here only 2 alternate bondings. NH2 over here at the 6th position, NH2 over here or amino group here at the third position, or sorry, second position. And here you have a hydrogen, and but there you have an amino group. And over here you have an amino group, and here you have a carbonyl group. Okay, so let's move on to pyrimidines, and I think I'm done with. Okay, forgot to mention this hydrogen over here, no hydrogen over here because there's a double bond. There's no double bond. So that's just the logic, and you just have to understand. Okay, if there's a double bond here, I know that nitrogen is sharing its third electron with carbon and both the carbons. But since there's only two bonds over here, the hydrogen, there has to be a hydrogen by default. Or if there was any other functional group, well, since there isn't, I've already laid out all the functional groups. We know that the only option is a hydrogen. And if there was a carbon, we would know it's a carbonyl group that would be there. So that's just the, and this is just essentially basically some basic chemistry. And if we have a strong understanding in basic chemistry, and this would become a piece of cake for us. So let's see, you have a carbon. Okay, so let's start over here. Let me actually do all the double bonds first. So you have this, you have this. Okay, so that's site. Okay, oops, I'm drawing. Okay, so this is actually cytosine. They all have a double bond. Okay, they all have a double bond. They all have a double bond. Like that, and here. But the difference lies that cytosine, and this is actually not that. I thought that was cytosine. So cytosine has two double bonds, and they're alternating. Okay, while as thymine and uracil do not. And actually, I should have drawn thymine over here so we can easily compare at the end between them two, because they're the ones that convert our, sort of switch in between DNA and RNA, but it's fine right now. So the thing is, cytosine is the only pyrimidine with having two double bonds. And remember, here we don't even have a third double bond, just like in the general structure of a pyrimidine. So that's pretty weird. That's pretty odd. So yeah, so we have two double bonds in cytosine, one and thymine, and they're the same positions. Okay, so now I think that's it for the double bonds, and let's move on to the hydrogens and the functional groups. So remember, like I showed you, hydrogen same for all. So that's one difference. Remember, we had four differences here, and this is our first difference. And now let's see. Okay, so there's a hydrogen, which is same. Okay, so it's let's see, yeah, so it's 2 plus 1 3 plus 1 4, 2 1 3 1 4 4. So it's these carbons are done. This nitrogen is done with it. Okay, and this nitrogen won't bond to anything because it's already sharing 3 valence electrons. And let's see, that's it. So yeah, so now the carbons in the position, so this is position 1, and we go counterclockwise 1 2 3 4 5. So in the position 5 of pyrimidines, there's a big difference, and we'll try to now explore that difference. The difference is that there's a hydrogen here, there's a hydrogen here, there is no hydrogen here, there is an entire methyl group or a methyl group. So CH3 actually. There's a CH3 here. And remember, that is actually the only difference between thymine and between uracil. That's the only difference that's there. It's the CH3, the methyl group. Okay, so now we know that the only difference between thymine and uracil is that uracil has a methyl group at the 5th position, while as thymine doesn't. It has a hydrogen. That's the only difference. The rest is the same for thymine and uracil. Cytosine has all the differences between the other two. Okay, so now if there was a difference between cytosine and the other two, that's probably the only difference that would count in pyrimidines. So now the difference is that At the fourth position, you have a carbonyl group for uracil and thymine, but an amino group for cytosine. And the second position, they all have an carbonyl group. So it's interesting to note how a pyrimidines, thymine and uracil have both two carbonyl groups, while as cytosine has only one carbonyl group, and actually instead of the other carbonyl group where it should be, it has an amino group. And I think that's it for the differences, and that's it for the structure. There seems to be no missing. Let's just confirm ourselves. So let's see, 4 bonds. Okay, 2. Oh, it's missing one thing. Yeah, it's supposed to be a hydrogen over here. And so same thing goes here. Okay, because I had a feeling they were missing something. So let's just confirm again. 4 bonds, 3 bonds. Okay, 4 bonds with the hydrogen. 4 bonds. 2 plus 1, 3, 4 bonds. Remember, the hydrogen is also an extra bond. It's bonding with the hydrogen. 4 bonds, 3 bonds. That's 4 with the carbonyl. 3. That's 4. Let's see over here. This is 2, 1, 1, 4, 2, 1, 1. Hydrogen 3, 4. So let's see, 3, 4. Yeah, and yeah. Okay, so we're good. So that's it. And lastly, so we have understood there's around, I think, 3 differences over here between the positions of the functional groups of the amino group and the carbonyl group and the methyl group. There is a methyl group in uracil, no methyl group here. There's an amino group here, no amino groups here, and they all seem to have another carbonyl group. And there's also another difference, and the difference lies that the bonding. Remember, the bonding is similar to the hydrogens present in the 3rd position of pyrimidines. Okay, so that's another difference, and here there's 4 starting difference. Okay, so these are the 4 nitrogenous bases and their chemical structures, and this together form an entire genetic sequence which help life to sustain. And because the genetic sequence or the genetic information helps to the cell to perform various metabolic activities and help sustain life. So on a last note, I think I messed up here. Well, everyone messes up, so it's fine. So it's actually thymine that has a methyl group, not uracil. But the rest is same. The rest is same. Everything else is same. Nothing has been destroyed. It's just that I actually messed up. I thought the uracil had a methyl group, but no, it's the thymine that has a methyl group in the 5th position, and the rest is same for them. It's just cytosine is all, you know, this has all these different types of differences. It has all these differences which uracil. So this is uracil and thymine don't. So thymine methyl group, uracil no methyl group. So T methyl and U no methyl. So since uracil doesn't have a methyl group, uracil, the IUPAC ID is, I'm going to use this red, demethylated, because there's no methyl, or it has been removed from thymine during the conversion or during the process of synthesizing uracil. Uracil is called demethylated thymine, and thymine actually is called 5-methyl, and 5 is just the position. And if you have some understanding of basic nomenclature, this is the rules that we use. So maybe that big font, 5-methyl uracil, because it's basically uracil, and uracil with a methyl on the 5th position, giving us thymine. And thymine is demethyl, and uracil is demethylated thymine because you have a thymine, and on the fifth position you remove. So if this was a CH3, you remove it, and you end up, and you just add a hydrogen, and you get no methyl but a uracil. So that's it for the introduction to DNA, what DNA is, getting a basic idea, and understanding in great detail about nitrogenous bases. And in the next video, we're going to talk more about the other two components, which is the backbone. That forms the backbone, and this forms the actual horizontal bases. So if you would imagine DNA to be sort of a ladder, so like something like this, like I showed you initially, the actual railing or the rails of the ladder, the actual backbone of the ladder, is composed of sugar and phosphate. I'll talk that in the next video, and I'll repeat that again. But the rungs, sort of the horizontal bases, are these nitrogenous bases, And the green is where the genetic code lies. And the blue, so the blue backbone helps give a proper structure and support to the entire DNA molecule, while the green information, so the green, not information, the green bases in the structure have the actual code. So that's it, and see you next time. Bye.